Hello, everyone, and a very warm welcome to our webinar titled Tired of the Status Quo? Discover the New Era of Purification with BioEcho in Admera Company. My name is Grace Box, and I am the Associate Director of Business Development and Marketing here at Admera, and I'll be your moderator for today's session. And I am absolutely thrilled to see and have such a diverse and engaged group joining us today. This webinar, we will be exploring a truly innovative approach to nucleic acid purification by BioEcho, who recently joined the Admera Health family. We'll quickly go over who Admera is, and then I will formally introduce Dr. Bürfent to you all, who will take it from there. I encourage everyone to please participate today by submitting any questions that you may have during the presentation. You can do that by simply typing into the Ask a Question box and clicking Send, and we'll answer as many questions as we can in the remaining time at the end of the presentation. And if you're also having any technical issues, go ahead and submit those questions in the same box, and we'll do our best to address them. So let's begin. So for those who are new to us at Admera, we are a service provider for advanced genomic and bioinformatics services. We leverage our expertise in next generation sequencing, which lies in our clinical foundation, as we are licensed in the US as a clinical lab. Our team at Admera, here you'll see this awesome picture. We're committed to providing expert scientific support and advanced laboratory services. And that's built on our passion for enabling groundbreaking discoveries. So we specialize in working with high risk and incredibly challenging samples. You'll find that our counterparts and competitors in the market will not be able to take on a lot of the samples and deliver these types of robust and reliable data that we do on our end. Our integration with BioEcho's innovative purification solutions, it really demonstrates our commitment to continuously strengthening our end-to-end offerings so that researchers like yourself or those who plan on getting into research could access these really high quality data from your samples. So as I mentioned, back in 2024, last year, really marked an exciting milestone for both Admera Health and BioEcho with this strategic joining of forces. This partnership is a significant enhancement for both of Admera and BioEcho, which allows us to elevate your nucleic acid workflows in a more comprehensive way. So by bringing together Admera Health's established expertise in next generation sequencing services and BioEcho's innovative and environmentally conscious purification tech, we're creating this super powerful synergy. So that means for you, you'll benefit from a faster, higher quality workflow, which leads to incredible results and also being kinder to our environment. So building on this powerful synergy, at Admera Health, we truly offer an end-to-end next-generation sequencing and bioinformatics service. So that covers every stage of your next-gen sequencing journey, as you can see illustrated here. Usually it starts all with extraction, as we've highlighted with our enhanced capabilities, now including BioEcho and their innovative technology. And then moving through the workflow, we offer a suite of services, genomics, epigenomics, transcriptomic services, and those who are interested in the broader gene expression, splicing, gene fusion events, what have you. We offer optimized protocols for incredibly low input and challenging samples. And then the more popular service nowadays is single cell and spatial transcriptomics, really diving into the cellular heterogeneity and disease mechanisms powered by your 10x, Parse, Takara, single RON, Stomics, and we also have a lot of customizable pipelines for you. And then any researchers who've already prepared their libraries, we offer a sequencing only option with leading platforms from Illumina, PacBio, Complete Genomics, and has incredibly high throughput capabilities. So finally, you have all this immense amount of sequencing data and you kind of don't know what to do with them. Our expert bioinformatics team, we provide this incredible comprehensive analysis across all of these data types. So this integrated approach with Admera and BioEcho's expertise at this critical stage here at the extraction stage really allows Admera to be your seamless partner for all of your NGS and bioinformatics requirements. So we're going to dive in deeper into the exciting science behind BioEcho's new era of purification and how it can directly benefit work. So I am super delighted to introduce our expert speaker, Dr. Benedikt Bürfent. He is our Vice President of Operations at BioEcho Life Sciences. His journey began with pioneering Ph.D. work in immunogenetics at the University of Bonn, followed by impactful postdoctoral research and a junior group leader position at Philipps University Marburg. He focused on the intricate interplay between genetics and gene expression and immune responses. So his deep understanding of the complexities of biological systems, coupled with his drive for better nucleic acid purification, led him to BioEcho. So again, I encourage everyone here to please participate by submitting any questions you may have during the presentation, and we'll simply answer them at the end of the webinar. So, Dr. Bürfent, the floor is yours.
Grace, thank you very much for this very kind introduction. I would also like to welcome all the attendances here for this webinar. My name is Benedikt Bürfent. I'm Vice President Operations at BioEcho Life Science, located in Cologne. And today's topic is to talk about nucleic acid purification. And interestingly, this is a quite important step in your experiment, in your study, and even more important if you would like to run large-scale experiments and studies. And BioEcho evolves a new era of purification, and I would like to introduce this to you and to highlight how important it is to choose the correct purification method to reduce bias in your system and to be aware what you will receive. The agenda for today is the company introduction. I will talk a little bit about BioEcho itself, the techniques of nucleic acid extraction. So I will give you an overview about different kind of extraction method, and then also give you a reason why BioEcho evolved and why Echolution, the technology of BioEcho, is such important to close a recent gap in nucleic acid extraction. And I will give you some examples where Echolution can give you an enriched biological insight and about the competitive advantages. Also, a very important today's topic. BioEcho was founded in 2016 by biotech pioneers, and they started in a basement as it should be. And they started also in writing patents and to evolve a couple of different extraction kits for different segments. BioEcho is located at the BioCampus Cologne here in Germany, and I'm very happy that I have a full team and experts in molecular biology from the science, also from the industry, and we have a couple of next generation sequencing experts also on board. We are passionate about sustainability and simplicity, so that means we would like to remove unnecessary steps, unnecessary chemicals, and we would like to improve the sustainability, meaning to reduce the carbon footprint of such an nucleic acid extraction. We provide not only kits, but also application support. So if you have questions about an extraction kit, you have questions about an experiment, and you would like to use and get aware of which benefits BioEcho could have, we also provide custom solution and services, especially for the field of breeding, biopharmaceutical industry, and also biomedical research. Nucleic acid extraction is a question of sample exposure, so the release of nucleic acids from a sample, and beside also the purification, so the recovery of a sample and to make the sample clean that you can use it in your downstream application. And of course, as you may recognize, both Quest 1 and 2 Simultaneously, very important, you need to have a very efficient lysis and also a very efficient purification so that in the end you can work with your sample and that you can have a good analysis. Sample exposure means that you maybe have a disruption of a tissue, you have a lysis or a disruption of a cell, and then this leads to the step of homogenization. The main task is to release nucleic acids from a sample. The challenges are a sample or a tissue could be hard to lyse, it's fibrous, we have a specific fragility of a nucleic acid, for example in terms of FFPE. We have to deal with nucleases, we have to deal with other compounds that does not really want to make the nucleic acid available. The biodiversity requires a different kind of different methods of nucleic acid extraction. Shown here, we have a various sample input. We have crops on the one side, we have biomedical samples with tissue and cells, but we also have something which is a liquid tissue like blood. And all they have specific requirements in regard to a sufficient nucleic acid extraction. For this, you can use chemicals like detergents, organic solvents, chaotropic salts. The most knowledge is available in science, and also you can prepare the lysis and the purification method by your own, but you can also buy commercial kits. Mostly they contain proteinases and other enzymatic activity reagents like lysosomes, simulase, etc. You can also work with chemical interactions like freeze-thaw cycles. You can beat your samples to homogenize and to open it, and also by sonification, but this is more prominent to proteins. Of course, there are a lot of pros and cons to different approaches. Here shown is the different kind of approaches and pros and cons for chemical, mechanical, and also enzymatic. The pros for the chemical sample precipitation: this is fast, this is more or less universal, but maybe the most of you know that this is quite hazardous and that you have to deal with PCR inhibition. The very prominent example for this is trisol. If you run an enzymatic-based workflow for sample digestion, it's quite flexible in respect of sample input and time, but it's also quite for the sample specimen specific. So if you're dealing with a tissue or a plant sample, it's somehow different. The cons are it's time consuming. It needs time to digest the sample. It's also expensive because if you use proteinase K, this needs to be delivered, and this is a quite expensive part in nucleic acid extraction. And of course, you need infrastructure. For example, a temperature incubation of your sample, and this needs power. This needs also a laboratory instrument. On the mechanical side, sample disruption. This is very powerful, this is very fast, true, but the cons are it's complex and it can lead to fragmentation. So if you want to have long DNA fragments to do some fancy HiFi sequencing or long read sequencing at all, you will need to run into problems with fragmentation or mechanical sample disruption. You can do this by beads and ultrasound, and interestingly, there are also some protocols in between. For example, first use beads to make a sample disruption of, for example, a plant sample, and then, moreover, work with an enzymatic digestion to digest the proteins and to make the nucleic acid available. The second twist is the purification step. This step is important to recover the nucleic acid from a sample. So you have everything free in your sample, but you now only want to have the nucleic acid. And this harbors also two important steps. First, the inhibitor removal, and secondly, the recovery of your nucleic acid, so that you have a nucleic acid in a quite high purity. We talk about to rescue our sample. The challenges are here, obviously. This may contain contaminants or inhibitors that may affect your downstream application, like PCR, library preparation, and other workflows. You also will have the risk of losing nucleic acid. Please aware, first, everything is opened. You have digested, you have disrupted your sample, but now you need to recover, you need to rescue your sample. And of course, you can also introduce some kind of bias here. You have active nucleases. If you're working with chiotropic salts, this is not a big problem at all, but if you're working with aqueous conditions or phase separation, this may be initially introduce a problem in the reduction of what you want to rescue. And this is also often a couplings processing to bind something to a bead, to a membrane, to recover it from a bead or a membrane, to wash it, to wash inhibitors away, and afterwards to get back what you initially bound to your surface. It's a quite complex challenge. By this, I would like to introduce you to the overall purification techniques. They are currently available on the market. Here it's quite important to say that the purification techniques introduces bias effects on your downstream analysis. Afterwards, I will show you example how we can, to our current knowledge, describe this phenomenon. The first steps toward a larger scale nucleic acid extraction happens in the end of the 17th with the silica matrix developed by Vogelstein and Gillespie, followed by salting out method by Miller in 1988. After CTAP by Doyle et al. At a time point where the germ wall fell down, we wanted to have, the community wanted to have a very cheap, but as you may know, a quite dirty method of nucleic acid purification, mainly for the area of plant breeding, where it's currently used. Afterwards, phenylchloroform, which is mostly known as the gold standard, for example, for RNA extraction, were published, and in the same year, the magnetic beads by Trevor Hawkins. And interestingly, these technology inventions happened in the 80s and 90s, and afterwards, now for a couple of years, no new development happened. First, I would like to start with the most known, probably most known nucleic acid extraction method, the liquid-based nucleic extraction, also called phase separation. First of all, you have your unique tissue or unique sample type, and you would like to extract your nucleic acid. After phase separation, you introduce a precipitation step. This then leads to the resuspension of the nucleic acid in your sample. Here, the pros are you have a high input and a high yield. You can purify both RNA and or DNA. And this is an inexpensive gold standard. The cons are obviously to make phase separation, you have to use highly toxic reagents. This is somehow complex to optimize and this also may lead to risk of contamination. And some customers also see that they have an inadequate purification step. Next, the magnetic bead-based bind, wash, elute. We also start with our sample. We disrupt, for example, or we digest. Afterwards, we introduce a binding step of our nucleic acid to a magnetic bead. And then we have to introduce a couple of washing cycles. The number of washing cycles are dependent on how dirty your sample is or how complex the matrix is in which this nucleic acid is located. After you run at least 3 or more washing cycles, you need a magnetic module to bind the nucleic acid bound magnetic beads to this magnet. You can remove the liquid, elute. After removal of the magnetic field, your sample in water, for example, also here in a reduced volume. The pro is here you have a highly flexible approach. You can nearly use this approach for nearly every sample type you know. It's quite easy to automate because also the magnetic step can be automated in a robotics. And you also have a good recovery nucleic acid. We're talking about around 80% or higher. The purity issue, the ethanol carryover, is a con of this method. If you would like to run a manual protocol or semi-manual protocol, it's tedious. High salts and ethanol required, and obviously a lot of step needs a lot of time. It's time consuming, obviously. And this method, because of a binding and elution, introduces a bias. To optimize magnetic bead-based purification, the equipment is somehow expensive. You need modules on your robotics, and the time and the power also is today quite expensive. The most or well-known solid-based nucleic acid extraction is the spin column-based. Every one of you probably already used such a kit. A couple of competitors of BioEcho provide those purification kits. You also start here with your sample. You disrupt your sample, you digest your sample. Then you introduce a DNA binding to a silica membrane. You wash, not only one, often 2 to 3 or 5 times, also in respect of how dirty your sample is. Then you have to discard the flow-through, which also contains chemicals. And afterwards, you have the elution step where you can reduce the volume, the initial volume, and where you can elute your sample in water and run your downstream application. The pro is you have quite high purity samples because you can wash all the things away that you don't want to have. It's quite safer to handle compared to liquid phase method. And you are able to concentrate your sample. Also like with magnetic beads. The cons is this is input dependent. So you have a quite small window of sample milligram you want to use. You have to deal with the chemical waste. You have a higher cost per preparation, and it's also time consuming because of the overall washing steps. As you bind your nucleic acid to a membrane, you introduce a binding and elution bias. If you want to collect also, for example, microRNAs in terms of an RNA purification, or London coding RNAs or others, you have to modify the percentage of alcohol also to get those fractions. BioEcho, as it was funded by previously 5 pioneers of other methods, as already shown, thought at the time point of 2016 that there must be something different. To come into the age of Alexa and artificial intelligence, we want to revolutionize and democratize the sample preparation and introduce a new method. This method is called echolution, and echolution overcomes ordinary disadvantages of other purification methods. We start with your sample. We have to disrupt, digest it chemically or enzymatic. Then you have your sample with DNA, proteins, and debris. You put your sample once on our echolution matrix. You have to centrifuge one time, and afterwards you have directly the ready-to-use DNA. You see, this is a one-step purification, which comes after the sample homogenization or disruption. Here the pros to overcome disadvantages are: we have a one-step purification. We do not introduce any kind of binding and elution bias. So all fractions that you expect in your samples, which should be present, splice variants, other variants, RNA species, all they are included. You have a high purity sample, as we remove all the inhibitory substances, also already shown by decreased CT values compared to our competitors. As you don't need as many tips and other things from your lab, it also reduces the laboratory costs. Your PI will be very happy not to pay for unneeded stuff in your lab. And we don't introduce inhibitory substances like chiotropic salts, etc. We work on so-called aqueous conditions. But also this method has cons. So we have to deal with a limited volume adaptation, as our metrics need a specific range. So we are talking about 50 to 100 microliters. As we improved the upfront lysis step, you are nearly in the same range like our competitors, with the concentration and the yield of nucleic acids as expected. All nucleic acid fractions are included. So if you don't want to have microRNA or other RNA subtypes in your RNA sample, this is probably not the best. So you can get the global RNA out of your sample. And for sample processing, you need a centrifugation or a positive pressure step. So you need somehow a force to push the liquid through the resin. But believe this 1 minute centrifugation step by only 1000 G can be done by nearly every centrifuge for spins and also for plates. As I initially said that we have only a one-step, single-step purification method, it's interesting which steps this may cover or really cover, because we don't introduce with our method any binding bias, a washing away bias, losing something that you don't want to lose, and also the elution bias. So are you aware how many of your nucleic acid is bound on your membrane after the elution step? With BioEcho, you process once, 1000 G, 1 minute, and you have your ready-to-use nucleic acid sample with the echolution technology. As you may anticipate, this can simplify laboratory workflows per se. Shown is the bind wash elute silica method. You start with the lysis and incubation step. You want to homogenize your sample. You want to get the RNA free. Afterwards, you bind it to your membrane. You have up to 3, 4, 5, 6 wash cycles, depending how dirty your sample is. Afterwards, you have to dry your column and to elute by water or low tris. And afterwards, you have to ready to use DNA. Beside, with the BioEcho ecolution technology, you also have a lysis and incubation step, which is improved. And afterwards, you transfer the sample to the already conditioned plate. Plate conditioning means that the volume of liquid which is present in the plate is removed previously. This can be done upfront and does not really affect the workflow at all. After 1 minute purification at 1000 G, you have the direct ready-to-use nucleic acid. Amazing. As I already said, we can save a couple of laboratory costs. Shown is an example of a cell culture RNA extraction. This is a case study we did with a customer last year. He ran 1000 samples in a 96 well format, and he automized the extraction on a usual Hamilton star. The disposal costs were €3.45 per kilogram. This is the pricing here in Cologne, the typical one. And we stated that the FTE costs are 22 euros per hour. Interestingly, as you can directly see, the grayer symbol are much larger than the ecolution one. And this is also true by numbers. So in this case, we need significantly less pipette steps and tips. We significantly could reduce the liquid waste, which relates to the disposal costs. We had less automation runtime, only 5 hours. So also with steps in between, in preparation, the robotics, etc., can also be easily done. And we can also reduce the associated costs per se in consumables, disposals, and also personnel. And somehow we calculated that you can reduce from nearly 12,000 euros to process 1,000 samples per day down to around 8,000 euros. So a cost saving of approximately 1/3. We anticipate that the ecolution technology covers all the today's needs in respect to speed, as we have a drastically reduced time required to compare conventional kits. In respect of recovery, that we have an unbiased recovery of nucleic acids. In respect of scalability, that we have less steps compared to conventional kits, and also in respect of waste. Also in terms of profitability, of course, reduced consumable costs and labor costs will affect your profitability at all. You are faster and you have a lower costs at all. And in respect of sustainability, which is also a very important step for BioEcho, with a reduced environmental burden and carbon footprint. And with some kind of kits, we can reduce up to 70% less plastics. So how can this product be placed in a market, and how do we anticipate its current trend in analysis of the different kind of market segments and also technology? Everything starts with a single analysis where the method is time consuming and you don't have such a big laboratory equipment to run all the experiment in parallel. So we see that over the time, also the next years with, for example, genotyping sequencing and other method, we have an increased technology gap from single isolations up to massively parallel extractions in, for example, a 384 well format. Ecolution can fill this gap. We see a trend in precision medicine, a need in genome editing, breeding, and biomedical research. You run bigger studies. They are funded by pharmaceutical industries, but also the government. And we see also an upcoming trend where NGS is more important, like in conventional qPCR. So the parallel sample analysis on larger batches, on larger scale, is more and more important. We can cover this gap by a more fast, simple, reliable, flexible, sustainable, and economical solution. As an overview, choose the best purification method for your study. Please be aware that you introduce somehow bias specifically for the method that you choose. We have the liquid-based one, where you have the phase separation and precipitation step and the resuspension. Here, hazardous chemicals are a main issue in this workflow. You have magnetic bead-based and silica membrane-based. Both work on silica surface, but you have this kind of tenuous washing cycles. You have to bind something and you have to remove something from the surface. Please aware that you introduce a bias. Our method try to avoid time-consuming washing steps and a binding wash elute bias. And for this, we expect that we can fill this gap and be the future purification method of the most of the customers. Now you would be interested in how our Ecolution technology can bring in more enriched biological insights. So to go away for a concept of I purified my RNA into I purified my RNA and I have a total RNA vision of my sample with a total RNA sequencing into a more global way, as this method tries to avoid biases during the overall process without removing something and has a more deeper lysis step also to collect all the classes and species of, for example, RNAs that you would like to expect. Interestingly, over the last years, not a lot of papers arrived who compared RNA isolation method with a downstream RNA-seq sequencing. Interestingly, RNA sequencing is in this case important to identify the RNA species, etc. But already in 2020, they were aware that the differences in RNA isolation method can substantially affect the relative transcript abundance. That the transcripts with higher abundance in the phenol isolated samples are strongly enriched for encoding membrane proteins suggests that phenol method better solubilizes those mRNAs. So we have a scenario where the RNA is not only present in the cytosol, but it's somehow in a cellular compartment enclosed and needs to be free. They also describe that they saw a decreased abundance of circuit nuclear transcripts in comparison to silica. Interestingly, this method does not really reveal nuclear transcript after the transcription process. And because of this, they were already more lowly expressed and thus more sensitive to appearing repressed. So they are not present, they were not recognized, and this can lead to false results. They also stated that the method of RNA isolation has a little effect on the ability to identify differentially expressed transcripts. So this means we have large population of RNA species where we can easily measure differentially expressed transcripts, but even in the case that we have a relative low transcript abundance, it's difficult to see whether there's an enrichment or not. Ecolution captures the strengths of both worlds. And this means in this experiment, this was subsequently done, we compared the extraction method of the ecolution method of phenylchloroform, but also to MEC beads, which are very prominent on the market. On the left side, as one of our first experiments in regard to this, we saw that the ecolution, interestingly, is somehow similar to a phenylchloroform extraction. So cluster R describes a very high similarity in respect to cluster B. And as we run a principal component analysis, we could describe that nearly 30% of the overall is different of cluster B to cluster A. So this describes how different this cluster is in respect to ecolution. On the right side, you see, interestingly, the same. You have cluster A describing the silica-based extraction method. And in cluster B, we have two subclusters, cluster B1 and cluster B2. Interestingly, they are not very similar, but they described a complete cluster. So as we see that we are nearly similar to phenylchloroform, we described that we also cover transcripts that they may be underrepresented in the MEC bead cluster B2. So we concluded that the use of particular extraction method produces a fundamental effect on the data, which is obviously, and this can already explain up to a 30% variation. Silica membrane creates a distinct cluster compared to phase separation, magnetic beads, and ecolution. So to bind something, to wash something, to elute something should really has an impact on your sample. And probably even if you also have the washing cycles on the MEC bead proportion, maybe the design of the MEC bead surface and also the elution is significantly improved compared to a silica membrane, but probably not on the same level like the ecolution technology. We also ask whether between the comparison of two groups, if there were somehow top regulated genes that they could be interesting for our community. So maybe you know CDK proteins in immune and tumor associated regulation. Probably you also need aware of MEF1, which is a mitochondrial elongation factor, but also MZF1, which enables DNA binding and is RNA polymerase II specific, interesting located in the nucleoplasm. So what is similar between left and right? On silica, we cannot detect CDK5 related transcripts, and we are not able to detect MZF1 in this case. Ecolution and MEC bead are enriched, so here the transcripts are present, but not in respect to the silica membrane. Interestingly, if you're looking to the mitochondrial elongation factor 1, we can see that also ecolution is slightly increased compared to MEC bead, but that somehow has an effect on the silica membrane. So we stated that in the overall context of the proportions of different kind of RNA species and how good we can open the cellular compartments to get all the RNA out of it in a more global manner, the purification method has a substantial effect on the extraction from RNA from cytosol and membrane enveloped complexes such as nucleus, vesicles, and mitochondrion. So this effect has an impact to discover immune and tumor associated targets, and this is very important to overall biopharmaceutical and biomedical customers. Interestingly, we also saw that the ecolution technology reveals an impact on therapeutic relevant RNA species. miR70HG is related to a long non-coding RNA, which is more prominent in the ecolution technology and all. And you can see on the left-hand side that there's a huge enrichment compared to MACBeat and silica. So we have, interestingly, an enrichment in a fraction that should be present on a MACBeat, but which is more and more probably unaffected compared to the silica-associated methods. Interestingly, for CAPN15, this may function as a transcription factor and RNA binding protein. This is not true, but overall we have a higher increase, stronger increase compared to silica, which is on a lower level compared to both other groups of ecolution and MACBeats. So the purification by ecolution reveal all RNA subtypes, and this includes also small length microRNA species to detect, in this case, highly therapeutic relevant markers. And we stated and anticipated that this is a more global picture of the cellular biology. And interestingly, if you compare all the RIN values of RNA integrity between all the three groups, we all have excellent RIN values. In the first two, we also have the microRNAs, so smaller 200 nucleotides lengths present. But interestingly, there is a difference between quantity and also the quality. And quality means a non-biased vision on the true cellular biology. We were somehow interested, beside the cell culture RNA scenario, whether this also may have an impact on our FFPE RNA species relating to tumor biology. And interestingly, when we made our first experiments, we were overwhelmed about the results. We did not really understand why we significantly had more unique mappers compared to silica. This was a really significant feature, also in comparison to normal and tumor pairs. We have less multi-mappers and a very, very small fraction of unmapped reads. We have less ribosomal RNA contamination compared, and also this directly relates to the number of genes that you can detect by BioEcho and the ecolution technology. So stated here, we have less junk and more meaningful reads, and this enables improved tumor characterization and pathway analysis. Of course, you have more high quality data in which you can currently work. And we also can relate this to a more cost-effective analysis of the transcriptome of tumors, because we have more uniquely mapped reads. You don't have so many waste reads, and you can reduce the cycles and also the number of reads that you plan to run for your sample on the sequencing system. Interestingly, we did this study not by our own, in a cooperation with Lexogen and Reference Medicine. As we now talked about the benefits regarding RNA, FFP RNA, and cell culture RNA, it's interesting that also the fact of scalability in case of plant breeding can also help to increase the knowledge here. Here's a side-by-side comparison. For time reason, I would also to talk briefly that we have a shorter protocol that we can avoid the binding and the washing steps, and that we are pretty fast in nearly 1 hour to extract our plant DNA. In cooperation with Sequel and GeneCov, we could identify that we have a broad range of applications or broad range of sample input material that our protocol can deal with. So soybean, tomato, and maize. We have a high concordance. So if we run triplicates, duplicates of sample, there is a very high overlap, less variation, and we also have very consistent insert size in the protocol. And as you already get aware of a principal component analysis before, we have really distinct subclusters of the varieties and the replicates itself. So a very, very good sample set. And why is this interesting? As we see the trend for an NGA crop genotyping, this can also improve science and also support the breeding industry in the future. And this is also true for an experiment with cannabis sativa, where we use the ecolution plant DNA kit, also with a DNA library preparation workflow from Sequel, and where we could also identify all the subclusters related to each other, that we have a very good insert size in mean, very good distribution, and where we could also align nearly 98 to 99% after sequencing. So as a summary, please be aware of the bias of nucleic acid extraction on your study and choose the best available technology for your experiment. So please be aware that this may affect what you see downstream, and if you run, for example, an NGS experiment or an NGS downstream application, this effect you will directly can see in this data set. So different methods have a different influence and importance on the experiment and should be really selected carefully. The more detailed the analysis are carried out today, the more important is a bias-free extraction method that becomes to obtain the complete biological image. And of course, as we introduce a new method, ecolution with a single-step purification method secures the biological insight and improves your downstream applications. It not only simplifies and made it scalable, but we also can directly remove contaminants, we purify the sample, and we directly can increase the data quality. But please aware, your sample also need to have a high quality. BioEcho empowers you to act more economically and to be more environmentally responsible. Thank you very much, and I'm happy to take questions now.
Thank you so much, Dr. Bürfent, for that insightful presentation and really demonstrating how traditional nucleic acid purification methods can be significantly improved upon using BioEcho's ecolution purification solutions. So we will move on to the live Q&A portion of the webinar. We have a question here asking, how do we separate DNA and RNA after ecolution extraction?
Yeah, so this is a very excellent question, because we have two different workflows. The one is protease-based, where we can directly purify DNA, and if we have less RNA contamination, for example, in some plant samples, we can see this. We can use an RNase in our workflow. On the other side, we developed also a new chemical-based workflow, where we can directly purify DNA out of our sample related to the first step of sample purification, and we can distinguish between the DNA extraction and the RNA extraction. And if you would like later then run, for example, an RNA sequencing, and you have to deal with, for example, a quite high amount of DNA because you have a low expression of RNA in your cells or tissue, for example, we can also introduce a gDNA removal workflow in a step in our workflow, so that you then in the end also have a pure RNA sample, and you can reduce the intergenic reads in your NGS. So this is quite also simple, like all the other workflows that we have in our portfolio.
Wonderful. So next question, even with centrifugation, can the DNA and RNA kits be automated?
That's a very good question. So the beauty of the method is somehow to reduce all the equipment that you need in the laboratory. So meaning to democratize also the sample purification means that you, by manual processing, you can easily run 96 well plates without the need of a robotics. The next step would be then to introduce a small liquid handler, like someone from Integra, SBT Labtech, only to transfer the liquid from A to B. Then we have the possibility also to make a semi-automation on an Hamilton Star, Beckman i7, all the bigger machines, where you then have a full deck layout, but the centrifugation step is then off deck. And this is often used by plant breeders, for example, because they would like to run the full deck and have the biggest performance. We also have the possibility to have a fully equipped robotics running. So an Hamilton Star provides a centrifuge on deck. You can introduce a centrifuge from Agilent to a Beckman system, for example. These are only examples. And then you can also run the protocol fully automated without any human interactions. And this is quite interesting for pharma clients. They request for a non-human interaction workflow also to reduce biases and to have standard lead times in the workflow.
Okay. Is there a nucleic acid kit that works better for NGS analysis? And what should I consider when selecting my extraction method?
Okay, this is now a hard question to answer. So I personally said, so I really like the RNA kits in our portfolio. As you can here, purify the RNA in 15 to 20 minutes. And you have a very high purity sample that you can afterwards run easily in an NGS workflow. And I already presented the data where we compared in one experiment against a BioEcho phenol against silica membrane, and on the other side, MacBeat ecolution against silica membrane. So I really like the RNA kits like cell culture and FFPE, and so also dealing with the need for more pure samples for crop science and for plant breeding. We also see that especially using the 384 well format in combination with the library preparation is also something that it's very, very interesting. The other kits are also quite good. Tissue DNA for animal breeding. So far, I guess the most benefits are already in the scalability of a kit in open words, and on the other side also to have an improved transcriptome analysis in the RNA kit. So these are my personal highlights in our portfolio.
Wonderful. I think we have time for one more question.
Yeah, I'm happy.
How is the recovery rate, and how well does it work with low input materials?
The recovery rate is between 80 to 90% to our current knowledge that we have. So we can get nearly everything out of a sample. If you have to deal with an older sample, if you have to deal with more fragmented nucleic acids, for example, fragmented DNA, the recovery rate can be slightly lower. So meaning that you, for example, can reduce the amount of very short fragments with our technology. So, but overall, the recovery rate is nearly on the same level like magnetic beads.
Wonderful. So thank you again, everyone, for your time today. I would like to also thank Labroots and Dr. Bürfent for today's awesome webinar. Before we go, I'd like to thank the audience for joining us today and for your awesome questions. Any questions that we were not able to reach today, and those were submitted during the on-demand period, will be addressed by Dr. Bürfent or myself via the contact information you've provided during registration. This webinar can also be viewed on demand. Labroots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. So until next time, take care, everyone, and have a good rest of your week.